Good afternoon, everyone. Had to start with a technical glitch. I welcome everyone who has joined us today, scientists, researchers, teachers, students from institutions around Delhi to the campus of National Institute of Immunology. Thank you for battling the unpredictable rains of Delhi and being here today for this open space on the much talked about BioE3 policy that expands us biotechnology for economy, environment, and employment. As the name suggests, this policy is all about making biotechnology work to boost the economy, sustain the environment, and generate employment opportunities in India. The policy was recently approved by the cabinet and has been in news ever since because of its potential to change the way we live. And we all have gathered here to unpack what BioE3 is all about. So without further ado, I request the Director of NII, Dr. Debashish Mohanty, to kindly address the House and also talk about the vision of this policy.
Okay. Good afternoon and thank you, Banya. So I'm really thankful to you all. So our Science Setu College students, teachers, my faculty colleagues and NII students for coming for this meeting at such a short notice. Probably you just were told yesterday or something. I'm really thankful to you all. So you must have heard a week back, Honorable Prime Minister and a cabinet meeting announced the BioE3 policy. It is biotechnology for economy, environment, and employment policy for fostering high performance biomanufacturing. So this is something not that often we have a science policy announced by the cabinet. And of course, this is something new. I mean, though, as you would realize, you are familiar with in some way or other, but how biotechnology is going to affect economy, environment, employment. So this aspect requires some understanding, thinking, and that is why we thought we should have this open space or panel discussion where the experts who are directly involved in some of this biotechnological work can talk to you. And I am really thankful to the panel for agreeing again at a short notice. Dr. Sam, Dr. Bist, and this one, and Dr. Gitanjali and Dr. Gopalan for this one. And so the type, why there is so much of excitement about this policy? As you would think, the technology has made changes. So probably industrial revolution came in at the end of 1700 or something. So then you had your steam engine came in 1850 or something like that. After that, you have electric bulb or something light came. Then the automobiles came, and something those of you, I think many of you students might not have seen. Some of my faculty colleagues or people of my age group would have seen how IT technology has made a change. If you go back to late 80s or 90s, if you think, if you have to book a railway ticket, so the number of probably each of us when we were in colleges or the hostels, when you have to go home, we almost stand half a day in the queue to book our train ticket. And probably none of you would have imagined those types of situation. You just book from your mobile, you are booking this one. So IT revolution has made a very significant change in our life and in many other ways, which we all see. And each of these revolutions, which we talked about, have made changes. So same way, this biomanufacturing is going to change our life in a very significant way. And that is why all this excitement, and that is what we should understand it better. So coming to biotechnology or something, so everybody, you will naturally think in the term of a vaccine or a drug or something. Of course, a vaccine is also a biomanufacturing process. But as you would know, biomanufacturing has much more than that. Any of you who would have read newspaper in Hindu, there is a article on biofuel that government has decided there will be certain percent of ethanol blending in the fuel petrol which we are using. And one of the technology is being transferred by some of the expert, Dr. Sam Esdani, who is going to talk. Some biotech work which was developed, biofuel work which was pursued in his group, he has identified some enzymes which can efficiently do this. And after announcement of this policy, one of the biotech company has come forward to take this policy, take it to the market and things like that. Same way, so each of you would talk up about something or other which we do in our daily labs, which is going to affect this one. And that is why this, and overall, this policy talks of is these are the things which we are talking is the these bio-based enzymes and chemicals, functional foods, smart proteins, precision biotherapeutics, which are these are a lot of antibody-based drugs, vaccines, other things, and precision medicine comes here, and climate resilient agriculture, and carbon capture, and how to convert carbon dioxide into some sort of a fuel or other type of thing. Those will be covered here. And in addition to that, there are many of the futuristic things like marine, how to use, we have a huge marine coastline, and how to make use of the marine resources through biotechnology, that will be also covered under this. And also the space research. And this Sunita Williams is in the space due to some reasons she is stuck there. And but we do not understand how it will affect the biological processes, your immune system or something. So ISRO has very active programs in this area. So these are the things which will be cost. And in addition to that, what is also envisaged, there are certain bioenablers. That is what the policy has involved. And AI is one of them. And the biomanufactured, like this one, these are what I would like to just emphasize is that when you do this biomanufacturing, like any industrial manufacturing, this will be done as a design, build, test, learn cycle. So like if a automobile is making, is first you design the car on the computer, then they this. So you would be now the biotech has developed enough for something. So now in this design as well as the learn cycle, you are generating a lot of data. So you are using the data very extensively, and that is where the AI is going to play. So bio AI, that is why it's a significant component of bio. This one of the bio enablers which Dr. Gitanjali is going to cover. And the thing is, the foundry is the word we don't hear in the context of biology probably. But if those of you who know anybody in metallurgy or mechanical engineering, every engineering college will have a foundry. Because if they want to design a product, they first make a prototype in a foundry. In the same way, even if you are cloning enzyme, like what is given example is a enzyme which you can more efficiently make this molecule a flavonoid and with a yield of 88 mg per liter titer. So this one has to test many different combination of promoter constructs and sequence designs or all these things. If you have to test 23,000 combinations in the lab, it's going to take several years. So that is where the biofoundry, where there are a lot of robotic processes are involved, is going to help. And these are the ones are going to be done in the biomanufacturing hubs. Like when IT technology came, you would think there were things called software technology parks you would have been listened about. What are those? There they provide all IT resources, internet thing. A company, somebody who wants to make a IT-based company can easily go there, use the resources, and without much investment, he can come up with this one. So what is envisaged that these types of resources, biofoundries and bio-AI centers or biomanufacturing hubs, this will be used. And the way you have the startups, like some of the startups would come up in the area of biotechnology. Already there are a large number of startups. There are around 8,000 startups in the area of biotechnology currently. And our bioeconomy has changed from $10 billion. Currently, we are at $130 billion, and it is going to change very significantly. And this is why, so I think today's discussion will, you can all, after the panel, you can ask, Banya will outline the things, you can ask all the questions and few other things just for the sake of excitement, the students, like the way things are going. This is a paper which is not published, but this is in bioarchive. I'm sure it will be published somewhere or other, where you are learning of self-driving cars, which Tesla is making. So people are already thinking or already making self-driving labs. So this is the automated robotic lab, or the automated scientist to design and optimize microbial strains for industrial production of small molecules like that. This one, so there are, it's the East thing, there are many 242 different compounds can be done in an entirely robotic enabled setup. So these are the things which are coming in future. So I would stop here, and Banya can take it forward. Thank you again, all.
Thank you, Dr. Mohanty, for showing us what the future can actually look like with such policies in place. Now, we will just move a little back. And what I will do is I will take you through the basics of what science policies are, why science policies are made, and just give you an outline of the BioE3 policy. This will be followed by the by short talks by the esteemed panel of experts that we have with us today, Dr. Shams Yazdani, Dr. Devinder Sehgal, Dr. Gopalan, Dr. Gitanjali Yadav, and Dr. Naveen Bish, who will break down the specific attributes, or rather focus on the policy that together lay the blueprint for its implementation. And then we will open the floor for questions, discussions, musings, anything. So yeah. So what actually a science policy is? It's rather a document which defines the direction of research activities for a country. Now, it's usually developed by the governmental bodies or the stakeholders like funders of the country in the country, or any agencies which have a kind of interest in science. Ideally, the government frames policies that are in nation's interest and in the people's best interest. A science policy may address many areas such as basic research, development of new technology, how these technologies can be commercialized. So, in a nutshell, a policy steers science into areas of importance and interest to society. Here I have put down examples of some science policies from the past in India. Now, yeah. Now the E3 policy that we are talking about today is one that has been crafted after very careful consideration of the needs of the future. Now, what are some of the needs that this policy addresses? One, to address the critical global challenges that we are facing: climate change, food security, health challenges. Next is to have a robust biomanufacturing ecosystem that can support innovation, especially in bio-based products. And again, the biomanufacturing will be a strong catalyst in boosting India's bioeconomy, which is projected to reach around 24,000 billion by 2030. So in order to enable this progress, a policy had to be put in place by the government of India. So the goals defined by this policy are: first, to drive innovation and solutions for fostering high performance biomanufacturing. That is one of the primary goals of this policy. To facilitate scale up and commercialization of bio-based products, intensify the entrepreneur setup of the country, and then to expand the cohort of highly skilled workforce with a surge in work or job creation. And then lastly, the most important thing is green growth, or to build circular bioeconomy by reducing, reusing, and recycling of waste materials. So these are the goals that we strive to achieve through this policy. And so now the need is in place. We have identified the need. The goals have been put forth by the policy. So the next step is that the policy lays down a framework of what we need to do to achieve these goals. And this framework is in the in the form of the strategic roadmap, where thematic areas have been identified where implementation will occur through different interventions. And also, they have laid down a strategic roadmap in the form of bioenablers that Dr. Mohanty just spoke about, which will be cross-cutting across all these thematic verticals that have been identified in the policy. So the policy here has identified why we need to implement it, what goals we are trying to achieve, and shown as the direction how we can reach there. And in the next segment of this meeting, what we will try to do is we will try to unpack the different thematic verticals under the policy with the help of the experts who have joined us today. First, I would like to invite Dr. Syed Shams Yazani from International Center for Genetic Engineering and Biotechnology to talk on bio-based chemicals, enzymes, and carbon capture and its utilization. Dr. Yazani leads the microbial engineering group at ICGB that works on leveraging biotechnology for biofuel production. Dr. Yazani.
Thank you. Thanks, NI director and other faculty member organizer for giving me this opportunity to speak in front of the very, very young people who are probably going to be a future bioenabler. It's very important. So as I was introduced, I'm going to talk about, my effort on biofuels area, but basically it's all going to be using, enzymes and biochemicals. So let me just tell you why I started working on biofuels, or rather, why it is important to work on bio-based enzymes and chemicals. Just to give you some of the numbers, most of the energy at this moment today we are using, everywhere there is a need for energy. Any manufacturing you talk about, there is a need for energy for that purpose. So where is this energy coming from? Manufacturing is one part, another part is the vehicle. So where is this energy coming from? So almost all the energy is coming from the fossil fuels. And that is leading to almost 35 billion metric tons of carbon dioxide release in the environment. So basically, that means that every year, because of the human activity, we are adding new 35 billion tons of carbon dioxide in the environment. So you can think about the cumulative effect of this that is generated in the environment. Now, this is known that this addition is leading to rise in temperature, as there are many, many different agencies that have confirmed that there are rise in temperature, almost around 0.2 degrees centigrade per decade. That is the rise that is happening. And almost all countries now are responsible for this. You can imagine that China, US, they are the major contributor. But India is also quite, responsible for this contribution. But what is important to understand is what is the impact of this particular addition that is being made in the environment. About 10 days back, I just was reading this article in Hindustan Times. Probably many of you in Delhi must be getting this Hindustan Times. I don't know how many of you just scroll through it. It basically says that Nipah and Chandipura are just tip of icebergs. Recently, I don't know whether you heard about these, viruses name. They are being in the news because they are new virus, not much known about these viruses. People are still trying to understand what is the nature of these viruses like Chandipura and all that. Now, after COVID, these are some of the new variants that have appeared. Now, of course, we have faced COVID and we don't want this COVID to come again. But the matter of the fact is that due to, very, very thin, distinction between the wild animals and the human, there is the boundary is almost negligible because we are actually, intervening in their ecosystem, and they are actually trying to find different ways to deal with or cope up with this. Looks like there will be a, what is most dangerous is that there are almost 10,000 different varieties of viruses that have abilities to jump from animal to human if this climate change is keep on happening. So that is a very, very alarming situation. And this slide gives you a little more information about why this is happening. The slide I took from CDC Atlanta, the Center for Disease and Prevention Control in Atlanta. They basically indicate that almost all the problem that today we see in the world is actually coming from climate change. So what is basically happening is that because of the rise in the carbon dioxide level, I told you that 35 billion tons of CO2 we are adding every year. There is a, a rise in temperature, there is a more extreme weather, there is a rise in the sea level, and that is actually leading to changes in vector ecology. Those vectors like mosquitoes, those type of mosquitoes which were not there in Delhi earlier, would you see that now? Earlier, we never used to have a chikungunya. Now you see chikungunya. In about a decade back, probably dengue was very new to at that time. Now you see a very prevalent cases of dengue. Now these are all happening because of this differences in the vector ecology. And of course, there are many other factors that are leading to a very, very extreme disease condition. So what you would realize is that after seeing all this, that probably prevention is better than cure. You know, do you want to just keep working on trying to solve one, you know, uh, disease at a time, or do you want to solve a larger problem? Do you want to really think about how to not let the climate change? Well, India is actually ahead in this area, especially trying to see how we can resist the climate change. There are many, many initiatives. And initiative really started with the Paris Agreement. And India is one of the countries which constantly trying to see if this agreement that has been made, we are reaching there. And one of the things that India has said, although many developing countries have said no, that we can't commit to net zero carbon emission. But India said, well, we will commit. And that's what it has done. They said that we'll commit to net zero of emission of carbon dioxide by 2070 and, reduce emission intensity of GDP by 45% by 2030. And how will they do that? There are many, many different programs that we'll do. But one of the ways will be to blend petrol and diesel with the biofuels that will reduce carbon dioxide. And ethanol blending has already been achieved by 10%. So we already replaced 10% of these fossil fuels with the biofuel. And that was done in 2022. Now the target is by 2025, can we replace it by 20%? And that's the goal that we all are looking forward. The only problem is so far, these blending had been done mainly from the first generation biofuel. I'll just touch upon it. And can we do, more than 10% by using some of those agricultural residues, like for example, parale that is being burned hugely, I mean, largely in the north area. Now, can we make use of those parales by producing fuels? And that will also give income to farmers. So you already heard about BioE3 policy. I'm not going to go deeper into it, but it's basically, it is all in line. When we have an environment in the middle of E3, that means government is so much serious about protecting our environment. And that is very, very key. That is the root of the problem. And therefore, whenever we think about making anything, producing anything, you think about environment first. So biofuel, as I said, is very interesting molecule because it's carbon neutral. You produce carbon dioxide at the same time, you assimilate that through the plant, and then, for example, these are cornstarch, sugarcane. Those assimilated products can be used to produce biofuel like ethanol that can be blended in the petrol in the car, which will further take the carbon dioxide. So there's really a carbon neutral. There's no additional carbon dioxide that are released in the environment. And that's the first thing you want to do. You certainly do not want to add any carbon dioxide, but at least you are not adding any additional carbon dioxide. If that much you can do at this moment, that's a great favor to the environment itself. Now, so far, as I said, the biofuels are coming from first generation biofuel, and these are mainly bioethanol, that is a microbial fermentation of sugar, or biodiesel, this is transesterification of oil. But they are mainly made from the sugar food sources, such as sucrose, starch, or vegetable oil, which is not considered to be a viable alternative for a country like India beyond a certain point. So therefore, we need to work on something like we call them as an advanced biofuels as against the conventional biofuels. The major difference between advanced like second generation and third generation is that they are non-ageable portion of the biomass, like parali, which has been burned. So once the grains are out, those non-edible portion, if we can make use of those for a biofuel production, that is really what we want. Now, how do you take fuels out of those parali which are burnt at this moment? So basically those agricultural residues, they are made up of lignocellulosic biomass like lignin, hemicellulose and cellulose. These are structural polymers. I'm sure you must have read it in your 12th class. These are basically a sugar polymer, cellulose and hemicellulose. So what we do is we do a small thermochemical pretreatment to get rid of lignin, which is phenolic compound. And then we add enzymes to saccharify, to hydrolyze those cellulose and hemicellulose to get a sugar. And those sugars are a mixture of C5, C6 sugar, which can be fermented to ethanol. Now, of course, there are challenges at every step. There are, technological challenges that we had to, come through when we make fuels out of it. For example, the enzymes, which cost around 30% of all the, expense, is very, very important to be worked upon. That's one of the hurdles why we don't have an economical second generation biofuel. Then there are other C5, C6 fermentation, and bioethanol is a very good fuel, the only biofuel in the market, but it's not as energy dense as the fossil fuel. And therefore, you want to work on other interesting products which are more energy dense. So I'll just quickly go through it. Basically, what we do is we actually make use of what is called biofoundry. We have a partial biofoundry in our ICGB. You will hear more about it. Basically, it will help you to, kind of design, build, test, and learn cycle. And using this, you can make better product. So we start with, for example, if you have to make enzymes for, hydrolyzing those cellulose and hemicellulose, we really go to the soil and we take, microbes coming from the soil. And these are part of biofoundry like high throughput robotic colony picker, robotic liquid handling system. And we make use of these to screen for the best microbes that can hydrolyze those biomass. And we put that, and once we have taken that, then we ask question, can we increase the ability of this microbe to produce more enzyme and more efficient enzyme? So these are the two questions that we ask. So for example, in our assay system, we found that one of the fungal isolate is a very good enzyme producer. We sequenced that, genome sequenced. We also did protein sequence, and we tried to address the question, how we increase the titer. So what we had done is, during the genome sequencing and annotation, we realized that there are several repressor of enzymes, catabolite repressor, and there are several enhancer for enzyme. So we worked on deleting the repressor and overexpressing enhancer, as well as we have overexpressed some of the key enzymes like exocellulase and lipopolysaccharide monooxygenase. These are the enzymes that we characterize are the best suited for biomass hydrolysis. You overexpress them, and as you see, the enzyme titer has increased as we do a more genetic intervention. And eventually, what you have to prove, if you want some companies to be attracted, because more than 90% of the technology fails during the scale-up process. So we have demonstrated that the technology that we generated in the lab In ICGB, we have these two 500 ml and 20-liter reactor is scalable in the industry. We went to the industry and we scaled up to 15,000 liter scale to show the scalability of the process. Now we are currently tied up with the industry so that this technology is now available for the commercial purpose. The other thing that we do in the lab is we actually engineer microbe to produce jet fuels kind of thing. The ethanol is one thing, but jet fuel is another area that you want to work on. And we actually engineer microbes. And interestingly, some of those compounds that you generate during your manipulation of microbes to produce jet fuels, they are also useful for the cosmetic industries, where we are working with some of the cosmetic industries. For example, cetyl alcohol, both jet fuel and cetyl alcohol, they have a very close pathways, and we are working to produce cetyl alcohol, which are actually present in almost all facial cream that you will see. Our engineering of microbe that basically was like 3 gene addition and 8 gene deletion led to a very high titer production of the cetyl alcohol and jet fuel. So another molecule that we started working on, with the help of industrial funding, this is called 1,4-butanediol. This is another bio-based precursor that you can use to make many, many different things that are currently being made via synthetic route, chemical synthetic route, or via fossil fuel route. So that's another product that we are very, very interested to work on. So these are some of the media picture, and like to thank my students for most of the work that have been presented, and I'll stop here. Thank you very much. I'm assuming questions will be later on, so.
Thank you, Dr. Yazani. Yes, questions we'll take at the end in our panel. And this meeting, this session is being live streamed on YouTube. If your friends want to watch it, who couldn't join us here today, you can send them the link, or you can ask them to just find us at National Institute of Immunology on YouTube, and you should be able to see the video. I would now like to invite Dr. Devinder Sehgal from the National Institute of Immunology to talk on precision biotherapeutics. Dr. Sehgal leads the molecular immunology group at NII, and his group is interested in developing monoclonal antibodies in the Streptococcus pneumoniae model. Dr. Sehgal.
So what I will be doing, I'll be describing a little bit about the lab and giving you an overview of how the precision medicine comes into play. So my lab has been interested in looking at monoclonal antibodies for a while now, and we have been generating a lot of different antibodies, mostly in the context of infectious disease. And we have characterized them in great detail. We have made more than 150 different antibodies. And today I will not be presenting much of the data in that regard, but just to give you a sense of what this precision therapeutics is all about. So I'll try and discuss that in a little bit. So very happy to be here, and it's an honor to talk about something that is not just at the cutting edge of science, but also something that will shape the healthcare and scene, as well as create opportunities for many of you in the years to come. Okay, so today I'll introduce you to the world of precision therapeutics and in the context of this new policy that has just been cleared and how this is likely to transform the field. Okay, so what exactly is biotherapeutics? In simple terms, it is basically a medical treatment or a process that is very specific to a certain medical condition, or it could also be personalized to individuals. So unlike the traditional medicine, which were often a one-size-fit-all solution, precision biotherapeutics tailors treatment in the specific medical conditions as well as to patients. And the idea here is that you target specific cells or molecules. So this overall approach maximizes the effectiveness and minimizes the side effects. So just to illustrate this point, so what I'm showing up here is, you have different cell types shown in different shapes, and you the so we have different cells, and traditionally what has been done is that you have a treatment which symbolized by way of this uh, small bar, and this is a method that will in fact not be able to distinguish between different cell types, and you also have some cells which are kind of transformed or they are different, and on and on it is something which will work like generic stuff. So you also have a situation where this is another treatment that is there, which cannot tell which binds to all the liver, the heart, and kidney cells, and we have some cells which are different. It would not touch. Let's assume they are cells which are unhealthy or malignant, for example. However, if you have, there are strategies by which the precision therapeutics, which will identify only the liver cell which is gone malignant. All the other cells are not picked up. Okay, so this would be a case for a precision therapeutic, and uh, so a great example of precision therapeutics is our monoclonal antibodies. Okay, so this is a type of biotherapy that will revolutionize how we treat diseases like cancer, autoimmune conditions, and some infectious diseases. Okay, so just imagine a situation where you can, your antibody is like a guided missile. It goes and hits the target straight away and gets rid of the cells which are damaged without um, giving harm to the healthy cells. So that's the power of monoclonal antibodies, and these are very specific. Okay, they are very specific, and also something to keep in mind is that just by way of numbers, the market for monoclonal antibodies in 2021 was valued at $186 billion and is expected to become 445 billion by 2028. Okay, so much of these developments in this area have actually been happening outside the country, and now with the policy in place, we hope that we will be able to do some of this within the country. So with regard to the policy, what it envisages is that it will make India a major player in global biotherapeutics market, and how would it help the country and people like us? So it's expected to be boosting innovation, and the policy aims to create specialized centers, as was mentioned previously, across the country that focus on cost-effective production of monoclonal antibodies. And by setting up these centers, we can enhance research and development, R&D activities within the country in this area. And this, of course, would mean that there's going to be more innovation, more breakthroughs, and as well as cutting-edge treatments being developed within the country, which will be useful for our countrymen as well as for people around the globe. And one of the other expectations that we have from the policy and is fostering entrepreneurship, and with the focus on biomanufacturing, there will be a surge in the opportunities for startups and new ventures in the area of biotechnology. And so imagine a future in which you and your peers could be leading a startup that is developing the next big thing in precision biomedicine, and this policy is creating that ecosystem that will perhaps help it for it to be realized. And of course, with all of these, there's going to be expansion in the job opportunities that would come by. And the emphasis on the precision biotherapeutics will create a significant number of jobs, especially for those who are still in R&D work. And whether you are interested in science, engineering, or even business, the growth in this industry means that there will be opportunities for various fields from lab research as well as management of biotechnology entrepreneurship. So, essentially, in terms of India's strength in research, development, and manufacturing is making the kind of environment that is important for capturing this India's share in the global monoclonal antibody market, and the rising demand in targeted therapies, increased public awareness, and better healthcare policies will fuel this growth. And with a growing elderly population and increased burden on chronic disease, the need for precision medicine is only going to increase. So in short, what we are saying is that the new policy is not just about improving healthcare, it is about positioning India as a leader in rapidly growing global market. And it's also about giving you the next generation of scientists, entrepreneurs, and the healthcare professionals, the tools and the opportunities to be at the forefront of this exciting field. So whether you are fascinated by science, driven by innovation, or inspired to make a difference in people's lives, precision biotherapeutics might just be the field for you. The future is bright, and it's an exciting time to be a part of this journey. So I'll close at that.
Thank you, Dr. Sehgal. These short talks are supposed to act as primer for the next segment, which is the open discussion on the policy, what it entails, and how the direction of the biotechnology research in India is going to be. So next we have Dr. Naveen Bish from National Institute of Plant Genome Research joining us to talk on functional foods, smart proteins, climate-resilient agriculture. Dr. Bish is a leader in agriculture biotechnology, working on biofortification, gene editing for quality enhancement in food plants.
Thank you very much, Vanya, for a nice introduction. And I welcome all the young scientists right here, researcher, I'll say, because most of them are from many colleges in Delhi. And then definitely we are all around plants, so they are very important. So I'll not speak too much about BioE3. I think already that job has been done to start to introduce more about BioE3, that is biotechnology for economy, environment, and employment. And what is it about? So I'll just put you some perspective and picture that India is among the top 12 destinations of biotechnology worldwide, and it is the third largest destination in South Asia, if I'll say, more specifically in Asia Pacific. And India bioeconomy, if you say, just to give you a picture, a figure on this, that it is an estimated about $130 billion in the year 2024, and it's projected to be almost about more than twofold by 2030 or so. So big challenge, big task for all of us to do this in the next 7-8 years or so. And now to address these national priority, as Dr. As it initially said also by Dr. Debashi, that there are many vertices which has to be looked on. And especially I'll be talking today more about the climate resilient agriculture and something more about the functional food or biofortification of foods, why they are very important as far as our bioeconomy is concerned. So when we talk about climate resilient agriculture, Sams has already done a lot of my job initially. We know that the global temperature is increasing, and on an estimate, we know that every century is about 1.3 degrees Celsius is the expected increase in the temperature. So what does it mean? So what it essentially means that we need to have more efficient agriculture in our land to take care of this global warming. Not only in terms of temperature, but we have higher carbon dioxide produced. SAMS give a very good estimate of the annual how much is the annual emission of carbon dioxide. And if some of you remember the value, it was about 35 billion metric ton. This is huge. Now just think about as far as plants are concerned. when we say so much of carbon dioxide in the at the global level, what does it mean? We need to breed crops which can sustain this higher carbon dioxide, because if you remember photosynthesis, it has to do something with carbon dioxide. We know there are two different C3, C4 by such photosynthesis, and we need to build more of, we need to utilize the CO2 more efficiently. The plant need to utilize these more efficiently. So to be the our we should aspire about higher yields or stable yield at least with the minimum carbon footprints. Now, when we say about carbon footprints, Parali is one of the best example. Are we increasing the footprint or no? Now we know that the rice stubble, which is used every year, which is now just the farmers are kind of, they are burning it. It's not, it could be a good footprint also for the, it can be a good circular economy for the agricultural land. But now we have to minimize all this footprint as far as the stable yield is concerned. Also maximizing the input use as efficiencies. Now, how to maximize the input use efficiency? For example, there are a lot of fertilizers which are being fed to the crops, including nitrogen fertilizers, phosphate fertilizers, various fertilizers, urea, DAP. Now, do plant use all of them very efficiently? No, the answer is no. Plant use almost about 10 to 20% of these fertilizers which are being fed in the soil. Rest 70 to 80% is going as a into our river bodies or down deep into the in our ecosystem, and we are not, and the plants are not utilized. So as a result, what we are facing huge environmental issues like eutrophication in lakes, and phosphate is a very good example for that. So we need to maximize the input use efficiency. More and more water, if the plant can utilize more water. The best example here is rice. We know that globally rice is the major staple food crop, and also in India, almost about 40% of our land is being used by rice for rice cultivation. Now, can we try to have a rice cultivars which can use this water, which has water use efficiency very high, so that you can grow rice in a small drop also, very few drops also, compared to what you are globally using so far, right? Rice uses a good amount of water. To even produce 1 kg of rice, you need more than 300 kg of water and 300 liters of water. So this is huge. Can we grow rice in even a very small amount of water use efficiency is very important. Now, transforming agri inputs, and we know that now there are many examples of transforming agri inputs. When we say agri inputs, those are the inputs which are used to fertilize crops, like fertilizers. Now, we know the best example here is nano urea, now which has been coming up to bring more productivity in crops. So that is why there is an urgent need to transform agri inputs also. And reducing loss is always a major concern, because our country being more of a subtropical country, we face a lot of pests and pathogens. So we need to reduce losses because of these pest and pathogen infection and infestations. So the goal of our country is to achieve climate ready agriculture, circular economy by adopting multiple approaches and integrating technologies which are coming in recent decades. So, for example, and what are the few areas I will try to convince you in a couple of more in slides, that what are these areas about synthetic biology and gene technologies can help us develop smart plants. For example, one example is to revamp the plant architecture. For example, the rice output, for example, the plant here, we need to engineer, as I told, we need to engineer more of photosynthesis. So we know that from a textbook that when we compare C3 and C4 photosynthesis, which are the most efficient photosynthesis in plants? C4. Why are they more efficient? Sorry? Yes, because even under higher temperature, even under higher global warming, C4 plants are going to perform better because they have higher, because they have higher photosynthetic rates, isn't it? And lower photorespiration. So they can utilize more of carbon dioxide even if we have more carbon dioxide footprint because of the fossil fuels. So always there are many approaches which are now ongoing in plants to bioengineer photosynthesis from C4 plants to C3 plants. C4 plants, some of the good example is sorghum or Yes. So from there, can we try to engineer plant C3 plant like rice to engineer this trait into C3 for C4 photosynthesis? So for that, we need to not only we need to engineer Rubisco for that kind of purpose for utilizing C4 photosynthesis, but also the plant architecture also has to be modified. So how can our smart agriculture can be done when we try to do all this stuff? So these are very big questions which need to be tackled down. These are very challenging areas which people are working, and there are with little success so far. But yes, this is something which is expected in coming future. Also, we need to also work on source sink relationship. We know that plants produce metabolites or important protein in one organ, and then they are transported to another organs. So this is called source sink relationship. But this conversion from source to sink is not always efficient. So we need to have information or the biology of these transport processes. What are the good transport protein, and how we can engineer those proteins to have better source sink relationship. That is new areas. And as Shams also talked about digestible fibers and for fodder. So especially lignin is very difficult to kind of digest enzymatically, because especially for biofuel, you need good lignase. So there are many efforts from ICGB, and also many labs in India are working to have more digestible fibers for fodder, for animal fodder. Agri-waste management is another area, as I told, for industrial needs, that how we can use parali for many various purposes. As many of you know that long back when we were kids, this parali, this agriculture waste used to be used for most of the shipment purposes. Now we are not using it for source purpose, because we are more of thermocol, polythenes to do this. So can we use agri-waste for our industrial purpose like biofuel? Can it be efficiently used? So Sam's give very good example for doing this. And there are for many other things which can be done in plant to improve the plant architecture. Also on system and synthetic biology or metabolic engineering of few uh, plant microbe association can be done. For example, there are good examples of some bioprotectants, like some of the biocontrol agent which can act as a bioprotectant for plants. If they are in symbiosis or they are in good interaction with plants, they can help plant preventing many root-borne diseases. Similarly, growth stimulant, also phosphate solubilizing microbes are also there. Nitrogen fixation, this is one of the very important area now, which is coming into very big way. Can we try, we all know that nitrogen fixation happens only in case of legumes. And that is why they are more efficient nitrogen, and they can utilize more efficiently from the soil. But can we try to do something in a crop plant which does not have this cycle? Like for example, rice, which we grow in a very big area of our land. Can we try to engineer nitrogen fixation there? That should be our goal. So all these areas are something which is rewiring our pathway from the soil microbes into plants, something which we need to work on both from the plant side as well as from the microbial side, which are there in the system. Now, when we talk about technology, genome editing is something a very big uh, invention. I think I'll say in this century, and how genome editing has shaped will be helpful in shaping the future ready smart crop. And I'll just give you some introduction about genome editing. As we all know, genome editing is one of the toolbox of modifying any genome at your will at a very precise location. So for example, you have a big genome sequence, almost, for example, a Arabidopsis genome, which is the model plant system, is about 120 MB sequence. This is so big. Now we have a technology which can pinpointly modify single base from that 125 megabase pair of the genome exactly at one certain position. So that is the power of genome editing. Now, so far before genome editing, people have been using genome modification using conventional breeding as well as using genetic modification. Now, genome editing is one of that big task from that one of the very improved way of modifying the genome. If to just for the introduction of the audience, there was a landmark decision by the government of India to exempt SDN1 and SDN2. That is, these are the two categories of genome editing, which is that is SDN1 is only when you have modification of only few nucleotide, and SDN2 these fall within the few nucleotide plants which are free from exogenous DNA. So when we do any modification, we always introduce some vector sequence. So we need to take care of that, whether that vector sequence is there or not. So these plants which are free from those vector sequence after modification, they can be exempted from biosafety assessment. So this was the rule which came in 2022 in 31st March. Subsequently, RCGM, that is regulatory body of the DBT, came into picture, and then they drafted guidelines and standard operating procedure through which the regulation of genome editing plants can be taken at the IBSC level, that is institutional biosafety level. And I will give you why genome editing and how genome editing is important. For example, as I told, through convention breeding, it takes almost about 5 to 7 cycles. This is a very textbook kind of picture, when you know that if you want to transfer any trait from a donor variety, which is, for example, a disease-resistant trait, into an allied variety, it takes almost 6 to 7 generations. So long cycle, long breeding cycle, almost 8 to 10 years. And then you have to handle a large number of population. One can also use transgenic breeding. For example, you can introduce a gene, but again, with the transgenic breeding, there is a major issue with the regulatory and biosafety guidelines. Now, through genome editing, we can do very precise manipulation at any given location, which are heritable. Once the mutation has happened, it will go into next subsequent generation. And the gene-edited plants are almost indistinguishable. That is, they are not, you cannot distinguish from any conventional breeding lines, like which can be generated through crossbreeding. So this is the power that this can happen in very fast manner. Even in 2 to 3 cycles, you can develop genome-edited plants which are free from any exogenous DNA. And I'll give you one good example from my lab, how we are using this genome editing technology for improving brassica mustard. So just to put some on picture, that India, our country meets almost about 2/3 of our edible oil requirements. So we are importing 60% of our oil from abroad, and that too mostly palm oil, which is not very useful. That is more of palmitic oil. Now it is putting a lot of burden to our economy. Almost about 1.56 lakh worth of crores of rupees is the burden to our economy because of import of edible oil. Now, Indian oilseed mustard, which is also called Brassica juncea, is the major oilseed crop which is grown in India. Now, over the last 30 years, almost 25 years, we know that the productivity of this crop is almost stagnant. Not only this crop, many of our oilseed crops are almost stagnant for the last 2 decades. We are almost about 50% of the global level. The productivity of our crop is almost stagnant. What globally is being produced. Now, if I'm giving you this picture here, you can see the productivity is almost stagnant. The yield per kilogram per hectare, we are almost, for the last 10 years, you see the picture has little bit improved, but not to a greater extent, and not at par with at the international level. At international level, this is almost about 3 ton per hectare, which is so high, almost 2-fold higher than what we are producing. Not only productivity, but even the quality of oil which we are consuming is also not very kind of health beneficial. Now, globally, canola quality oil is regarded as very useful oil. And when we try to improve canola quality oil, we need to improve we are improving its oil and oil cake quality by improving glucosinolate. This is one secondary metabolites which are present in Brassicaceae family member. So whenever you use brassica oil, you find a peculiar pungency on this oil, and it is because of the glucosinolate and erucic acid. A very high amount of these metabolites are not regarded as good. They are anti-nutritional. So the goal is to reduce these glucosinolates to a very low level in seeds only. But what is more important, that these glucosinolates are also important for plant defense. These are secondary metabolites. And if you try to reduce glucosinolates from the whole plant, then they are decreasing the plant defense potential also. So what our lab has recently done, we have used genome editing to make ideal mustard line, which is having low amount of glucosinolates in seed and very high amount of glucosinolates in leaves. And how we did it? We know that glucosinolates are synthesized in leaves, and then they goes to the seeds. So the glucosinate synthesizes all in the leaves, and then they are transported to the seeds by transporters. They are called glucosinolate transporters, GTR1 and GTR2. And there are almost about 12 glucosinolate transporter genes in Brassica juncea, which is Indian mustard. We use genome editing to edit to make these 10 homologs of these 12 GTR genes non-functional by making use of genome editing. We precisely edit GTR1 and GTR2 genes, and as a result, what happened, that the glucosinate no longer goes to the seed. So the seed has very low amount of glucosinolate, so it is good now for nutrition value, as well as the oil cake value, which is fed by the animals, poultry. This now it is more nutritious for both human and as well as animals. And very high amount of glucosinolates in leaves are now making plant more protection against pest and pathogen. So this is something which we have recently showed that genome editing could be very important tool, agricultural tool for biotechnological tool to meet some of the breeding objective which cannot be met by any conventional breeding or even genetic modification. So through genome editing, we have developed canola quality mustard, that is nutritious mustard with improved oil quality. And the seed meal is a very important protein rich oil cake for poultry and livestock. So this low glucosinate oil meal cake is also important will be useful for livestock and poultry feed. And now it can be exported globally also. And now as a result of higher glucosinolates in leaves, because otherwise we are not compromising on glucosinolates on leaves. So glucosinate in high leaves will reduce our dependency on pest and pathogen, as well as it will improve our yields also because of pest and pathogen. And so moreover, these plants are mostly SDN1, that is, they have only few mutations, so they have very least biosafety concern and higher public acceptance. And so Department of Biotechnology has contributed a lot in agro ecosystem, and to name few, this is the last slide, which shows that there are many call for proposal on genome editing, and this is one of the success story from any DBT institution, that how genome editing can help you breed crops which are nutritiously very well, yield better, and perform also very good under field condition. Similarly, they have released through DBT funded project, there are several crop varieties that have been developed through market selection approach. And the DBT is also running multiple mission mode projects leading to trait and gene discovery for climate and disease resilient crops. And almost there are many infrastructure strengthening is also happening, like speed breeding is the now next thing which is going up. At today's pace, one can only breed one can have only one generation in a year, but through speed breeding you can have 4 to 5 generation of every crop under controlled conditions. So you can multiply, you can have generation time very fast using these speed breeding. And we have speed breeding centers established in Varanasi, PAU, Punjab Agriculture University, Mohali, Nabi, and also we are just also coming up at NIPGR, which is just next door to NII. So there are many opportunity for breeding better crops using genome editing, or not only genome editing, but there are the basket is very big using genetic modifications also. And thank you very much for your patience listening. Thank you.
Thank you, Dr. Bish. Now we have Dr. Gitanjali Yadav from National Institute of Plant Genome Research, who joins us today to talk on BioAI hubs as bioenablers. Dr. Yadav is a specialist in genomics and structural bioinformatics with specific research interest in food security and ecosystem conservation. She's our complex network AI big data expert on the panel today.
Thank you, Banya, for that introduction, and big congratulations and compliments to NII for putting this conversation together. I think since the BioE3 has been announced, many of us are, including our PhD students, up to fir bhi college ke bacche hain. There are many people who feel ki what is this policy about, what's it doing? And of course, Banya and the director of NII, Dr. Debashish Mohanty, as well as my colleague Shams, Naveen, Devender, have already told you about the BioE3 policy. It includes innovation-driven support to R&D and entrepreneurship through various thematic sectors. And it will hopefully, we are envisioning that it's going to accelerate technology development and commercialization, eventually leading to greater skills and better jobs for all of you, by establishing biomanufacturing and bio-AI hubs and biofoundries and so on. So my business here comes in from the bio-AI hubs angle, because I'm a data scientist and I want to hopefully present to you or introduce to you what is this business of bio-AI and biofoundries that you have just heard or read about in the BioE3. Now, how many of you are aware of the United Nations Sustainable Development Goals? Kindergarten se hi aajkal school mein project milne lag gaye hain SDGs pe. How many of you are aware of how many goals are there, the UN SDGs? 17, yes, and 169 associated targets also, which are indivisible, where leaders of the world, all the nations in this was a landmark meeting in 2015, September, right? That's when everybody decided that all the nations together decided we are going to get rid of poverty and hunger by 2030. So the target of the United Nations SDGs was 2030 for these 17 goals. And different countries, all of us came together to say, yes, we are going to do something about it. And therefore, I'm very, very happy that this BioE3 policy for India allows people to, before that, let me just tell you that the United Nations Deputy General Secretary, Amina Mohammed, right? Last year, she, in the summit, and they keep having these summits. So last year in the summit, she said that the 17 goals over the last, now we are in 2024, we're very close to 2030, but how much have we achieved since 2015? How many of you think that, yes, out of the 17 goals, 10 are done or 16 are done? The truth is that for none of the goals have we reached completion. And for most of the goals, we are sitting at 50% short of what we had envisaged in the indivisible 169 targets. And so what the UN Deputy Secretary General said, that the scene that is missing here, and to make the SDGs happen, is technology. Modern technologies have got to be used by each and every one of us. And which is the major technology that is completely transforming all of us today? I would say it is AI, absolutely brilliant. And so each one of you, already your life has been transformed by AI. So why not your studies, your education, and your research? We are doing research. We've been doing this forever and ever, but at least with the BioE3 policy, finally, we are at the forefront of being able to drive something, including the UN SDGs. My own work, I work at the National Institute of Plant Genome Research. You just heard Banya introduce me. My work is primarily in food security, where we are trying to again use AI over genomics for addressing the photosynthetic capacity and improving the productivity, primary productivity of plants, essentially to also sequester carbon, green carbon. So black carbon in the atmosphere becomes green carbon, which is organic carbon. How? Through this reaction, the single cell reaction that can convert inorganic carbon to organic carbon through Rubisco, called photosynthesis. And we are trying to use this process in some of the most efficient organisms on the planet that are doing photosynthesis. So who are the most efficient organisms on the planet that are doing photosynthesis? Yes, yes. Who said algae? Absolutely. Marine phytoplanktons. Brilliant. We have geniuses here. So we are working with marine algae, and we are trying to address what is it about their photosynthetic process which can somehow be extrapolated into higher plants. That's my job in food security. And the second one we are doing is AI for climate change. And why is that important? Because of course, going forwards, the three major issues that the world has to face is number one, climate change. You have to deal with climate change. It's not a future problem. It's yesterday's problem. It's today's problem, as you've heard my colleagues talk about just now. The second problem that we all have is also mental well-being. I think with all of us, with the huge information that we are getting, the information data overload that we are getting constantly, have you noticed your attention spans have gone so chota? Mera fourth talk hai, ab tak sab so chuke hain, nobody can hear. That's because you don't have an attention span that will allow you to listen to what's happening. And so the world has already come to terms with it. All of your reels, I mean, this generation, your generation is all about reels. You don't watch movies, I suppose. You don't read books anymore. You're watching reels. And so therefore, we as researchers and educators have got to somehow change the way we connect with you. Hopefully today with NII, with these short talks, we're going to somehow get through to you and get to do what we. So I work on, so this is where Banya did not talk about what I have co-founded this global citizen science movement called Semantic Climate. And we perform AI over NLP, or natural language processing, to address or make climate data available to every citizen. It's not about making it available to researchers, students. It's about making it available to you, your younger sister, your great-grandfather, or retired local policy counselors, anyone. So what kind of AI? So AI can, of course, be used. AI over vision is one thing. Most of the AI that you know about, smartphones, Mera Photo, Tera Photo, and most of the smartphone apps that you use are about AI over vision, but AI is, of course, very heavily being used over text. right? So that is called AI over natural language processing. And the third one is AI over, I mean, there are many more, but there is another mechanism of using AI over sound. And sound bites or sound data sets. Can somebody think about how AI over sound might be useful? In the first slide that was presented by the director of NII to you, one of the things was marine biotechnology. So in the oceans, if you can record the sound bites of ocean organisms or ocean life, you could potentially be able to identify what are the, and this is being done right now by PhD students all over India. So how do we, with the AI that we do, how do we actually get the work that we're doing done? It is being done through knowledge champions and young people mostly. So what we have done, at least in my lab, in our research, is that we come up with reels. I should not say reels. I should not say attention span devices. What we've come up with is short projects. So we want interns to join us all the time. And a lot of research laboratories in India, at least in India today, and of course outside of India as well, are very welcome to the idea of some of the youngsters like yourselves joining us for short research projects, starting from 8 weeks all the way to 12 to 16 odd weeks, or even longer sometimes, to do some project. A student joined us recently, and he decided to look at some of the camera traps data that we had collected over a long biodiversity, climate change angle within Delhi, right across the road from NII. If those of you who have come here, badi mushkil se rasta mila hai, and Aruna Asafali Marg, if you cross the Aruna Asafali Marg, you're going to some place called the Sanjay One. In the Sanjay One, there are these things called golden jackals. Many of us don't associate Delhi to be a big, biodiversity hotspot, but India is a biodiversity hotspot, and cities even more so. And so what we have in the Sanjay One is something called the golden jackal. And so we have camera traps data. And one of my students who just worked with us for about 8 weeks was able to use the Facebook Softmax Python program of, that is an AI model, built on or trained on faces, all of your faces, because most of you, I suppose, have FB profiles or some social media profiles. And this softmax function can be trained to not just recognize people's faces or human faces, but recognize other things. And we try to test it. Within 8 weeks, this boy has been able to train that model on the camera traps data, which he cut into smaller pieces, like 2-second clips. And it can now recognize the golden jackal, separate it, distinguish it at 100% accuracy from the civet, which is also around there, little macaques, which are also in that, and not just from the frontal picture that you're getting from the animal. Camera traps can, trap anything, any movement. They trap side, tail, the ear, sometimes movement. And the AI is brilliant at doing this. Now, how could we use this now that it is successful? As a researcher, my question is, what else can I do with it immediately? One thing in the olden days, we used to always say, we will publish what we have and we will get some paper there. Yes, let's do that too. But the BioE3 policy says, make it an application, create a product, think about what you can do to make a change to society. Within biodiversity, India being a biodiversity hotspot, can you use AI to address the human-wildlife conflicts? Can we maybe use this trick that we've developed, or this success that we've had with the golden jackal, to be used for tigers? India's national animal is the tiger. And the tiger has issues such as, within the national protected areas, it strays outside. It strays outside, finds a village, finds some cattle, eats it up, and the villagers don't like it. They kill the tiger. The moment you kill a tiger in India, it's a national scandal. It's an international scandal. So what is it that AI can do to address this? Forest officers are having so much trouble in being able to predict where the tiger will go, which tiger might cross that space. But if we use the camera traps data, which is being collected from time immemorial, and we use geospatial imaging, knowing where the camera trap data is and where we can map this movement pattern of every animal. And what is the other thing about the tiger that you learned in kindergarten? It's got stripes. What's the thing about stripes with the tiger? Unlike the jackal, where we only identify the animal versus another species, what can you do with the tiger? It's a fingerprint. The tiger's stripes are unique to each tiger. If they can be learned by AI, you know which tiger is going where and doing what. And so you can predict and tell the forest department officials that my AI predicts that your 3 tigers are beginning to stray out. Let's keep them contained and let's protect them from having a conflict with the people staying around. So that's one thing. But I think I'm digressing here. Coming back, at the core of the BioE3 policy is this idea about building BioAI hubs in different locations. So we have this lab at NIPGR. DBT has 14 organizations. Every organization has the capacity to do a lot of informatics. So combining biology with computer science, with artificial intelligence to address questions that we've been forever addressing, but address them faster, particularly with the kind of attention span our future generations have. So we've been doing a lot of data intensive machine learning, deep learning, using the kind of neural networks that the brain builds. And that is why artificial intelligence is more of a biological computing. And so we do reinforcement learning, and we apply these to the kind of data I spoke about, which is in climate change, biodiversity, and food security. But what you can do, what now the BioE3 is thinking about is that you basically use BioAI. So the next step first is to get more of you to understand what is AI and use it on a day-to-day scale, maybe with small projects. Start joining some of us. Do it in your summer holidays, winter holidays. What is that thing? The Share holidays. Something you can do. You can do it sitting at home. This is AI is something you don't have to come to the lab and be at a certain. As long as you have internet, everybody has internet. My 8-year-old has internet. So as long as you have the internet and a device with allowing you to do some math there, I think you can run your own AI models, and it's not difficult at all. And so it's the perfect thing for people with low attention span. As long as you have something to think, you still have a brain remaining. Many, including my 8-year-old son, don't. But if you have a brain available to think something, you can come up with a new problem and how AI might be able to address that problem. Eventually, in the second stage, the BioAI is going to come into the picture, where the idea is to grow brain organoids or miniature biological AI. Okay, now where does that come from? What's the history? How do you store your data? Today, let's say you've got, what should I say? Yeah, let's say, I shouldn't say you have 10,000 genomes. Let's say you have 10,000 movies. Ab to koi rakhta hi nahi, of course, everybody is on apps. But let's say, so where do you save stuff? In the olden days, in our days, we used to have floppy disks. People laugh at the idea of floppy disks today. They don't exist. Even if you add them, nobody can read them. Then they came CDs, then tape drives. My entire PhD backup is on tape drives, jo aaj ki date pe koi reader pad nahi sakta. So all of that is gone down. So what is happening? We have very little, within time, modern technology is changing so fast that the storage, the data that you had is no longer readable or accessible. So what is the next step there? How can biology come in? DNA. You know how long DNA can store data? Thousands of years, tens of thousands of years, right? Fossils mein se nikal rahe hain. So can we use, so the idea behind this is, can we make biochips? Can we use DNA chips where you can store the information you want to store, and it will not be like a floppy or a CD or a tape drive that goes off. It will be stored in DNA because the information is going to be embedded in DNA, which is nanocellulose kind of semiconducting nanocellulose molecules, and they can stay forever. And so the idea of being able to use biological concepts to be able to, to first of all store data, or to address global problems, to address the SDGs, and do your own research is where the BioE3 is going to come in and make things possible for us. So with that, I think Banya had also said to say a few words about bio foundries, but I heard Shams has already presented this to you. Biology is an astonishing builder. Think about your little eyeball, the way things are built. Think about a spider's web. So what I was thinking was the kind of silk that the spider's web material is building. The spider has learned over millions of years what kind of, tensile, light, economic material to use for its web. Can we learn from biology to make better products, better things? And can that eventually lead to the complete transformation of the country with the largest number of people in the world? Like my son says, every Atwa Banda is an Indian. Do you know what will happen in the next generation? All of these Atwa Bandas will have their own kids, and every Agla Banda will be an Indian. Eventually, the universe is going to be. So the thing is that we have the strength in numbers. We have the strength of people. Let's use the strength of AI or bio AI to make, for a Vikasit Bharat or a biovision for India in 2047. With that, I think I will end my little speech. Thank you very much.
Thank you, Dr. Yadav. And now a peek into the future with Dr. Gopalan Sampat Kumar from National Institute of Immunology, who leads the chemical glycobiology group at NII and is exploring role of glycoconjugates in biological and immunological processes. But today he's going to speak on futuristic marine and space research.
Thank you, Banya. And thanks for everyone for attending here. I think we are overshooting the time. So I would like to keep it about 5 minutes and then open up the floor so that we can listen to the next generation and their questions. I'm sure you are all warmed up by now. And our director, Dr. Mohanty, has a knack of getting people out of comfort zone. And can you imagine? The maximum I have flown is about 30,000 square feet into the sky, Vistara or Air India. Okay. Maximum I've gone down into the ocean is about a little bit of snorkeling in Andamans. Okay. So I have a huge conflict of interest of not knowing enough about what I'm going to speak and not practicing those. But nevertheless, the principles of chemistry and biology are going to be the same, right? So we can still, this is futuristic, right? So if what I say is not happening in 50 years, you may not find me. Okay. So, so that being the case, right? So the goal here is futuristic is important because because people go on a cruise, right? You don't see your padosi for 6 months, 8 months. You're like, where did you go? I went on a cruise. Okay. So was Columbus, who went on a cruise, never found India, right? Isn't it? So that's futuristic for us. Are you ready to go? And so we might have sooner or later, where did you disappear for a month? Oh, I just took SpaceX and went up and I spent a month near moon. I was roaming around moon and I came back and intact. I didn't get burnt on the way back, isn't it? So that's futuristic research. We have to go there. And so is marine research also, like Gaganyaan, Samudrayaan, So we can measure the tallest of the limits of the universe, and then we can also deepest to the oceans and figure out what biology exists there, and also learn how much we may have spoiled it inadvertently or knowingly or unknowingly. Okay. And same is for space debris, right? People say, oh, we launched 700 satellites. Someone said, I don't know. Okay. Over time, if they work, it's fine. But when they fail due to a solar storm, who is going to clean up all the mess? So it's not just about going to space and having fun like a cruise. It's also about not affecting the environment. Okay. And there has been a ton of research. We are not the first one to get there. And so it is important for the next generation, and including me, current generation, to know how to approach this, how to harvest this without harming the environment, without harming the space. And that being the case, we are in a monsoon session, right? How many of you think that what if does it rain in moon? Yes or no? Yes. Okay, one yes and no. Everybody says no, right? If it rains in moon, then poor Sunita Williams, right? She doesn't have to struggle. I heard that when she went first time, her mother is a Slovenian, father is Indian origin. She took a samosa. Okay. But imagine we go out of town for 2 days and then, my flight gets canceled and we are like fish out of tank. Oh my God, all my plans got canceled. I need to get back to home. Here you have Sunita Williams stuck on a Boeing Starliner and they don't want to take risk of life. So they say, wait until January, big deal. Okay. So now imagine, what do you do? Okay. So, you cannot, you're like, oh my God, all my routines are gone. My flight got canceled. My train got canceled. This is ridiculous, right? We are imperfect, but here you are stuck on a space station. So that means you have to think about how to use these things positively. And they have a beautiful plan. The NASA announced, and they said, right now, according to, I'll come back to that later, that she and her partner actually trying to work on how to make fiber optics in space cables, and also how to grow plants. Okay. So those are the things that one has to understand why futuristic research and marine research and space research are important, because there is so much to learn. There is so much to apply for even Earth. It will have applications and to save against pollution. And one of the best thing I found was this essay by Robert Colson, like in 2001. This was an essay commissioned by Economist Shell World Essay Competition. 23 years ago, he has said this: given the already stressed state of natural resources throughout much of the developing world, it will not be possible for many of those countries to attain first world standards of living with industrial infrastructure as wasteful as the United States or other developed countries. No, it's not from a lecture platform to say that, oh, you guys cannot have it. No, please don't be as wasteful like those guys were. They did not care for 50, 75 years. Okay. And so the developing world simply cannot afford that industrial inefficiency or energy inefficiency. So at least we can make our own technologies that can be rivaling, that can be much superior to what others have done. We build on them, but we better them, right? So we have to better them in order to do that. And this was written in 2001, 23 years ago. Okay. And he also even predicts about doing, PCR amplification in your garage. And last year we had CRISPR-Cas9 kits being sold to houses in California and other places where you can easily run your own CRISPR-Cas9. You can run your own E. coli culture or a fungal culture. Like all of us culture lactobacillus, right? You don't need biosafety for culturing yogurt in your home. So the idea is that how can you manipulate lactobacillus or some other bacteria and say, oh my God, this will give me nothing but this particular chemical. Okay, so it's about doing that kind of a thing. And so Gaganyaan, I'm starting with a further. It's microbial biomanufacture will be providing integrated solutions, food for astronauts, biosensors, monitor astronauts' health from remote, austere locations in the future space mission of the country, and marine resources across the extensive coastline of India will be tapped to power the bioeconomy of the country. Okay. And one of the things that I found was actually there are many parallel efforts also happening in the Ministry of Environment. There is National Center for Sustainable Coastal Management, where people talk about mangrove for carbon dioxide sequestration. See, that's something you would have found, right? On the one hand, we keep saying, oh, Parali is getting burned, CO2 is getting to the atmosphere, and we have pollution and greenhouse effect. On the other hand, we also order CO2 cylinders for cell culture, right? I don't know how many of the dry ice, what is the consumption of dry ice in the country? What is the consumption of CO2 cylinders in the country? Are those people who are making dry ice, are they even connected to these burning people? So if we somehow connect it, we would have closed the loop. It's called loop closure. Okay, we are generating CO2, and then luckily we have these plants that someone said whatever CO2 we are exhaling, as we have been here for an hour, hopefully those plants will pick it up. Do you see a carbon dioxide linear molecule coming and sticking to the leaf? Oh, I see that. Okay, so we have to get to that kind of vision to be able to say that how can we close the loop? On the one hand, we cannot be burning. On the one hand, CO2 industries cannot be generating CO2 through other means. So that's another carbon dioxide capture. And then you look at plant nutrients and how land-sea exchange margins, that sea erosion and other things, organic matter assembly, and people want to talk about air-sea exchange of rate of gases. We think we make a lot of emissions. Even ocean is full of life, and suddenly gases may come out of it as well. So we don't know how those atmospheric interactions are happening. And microalgae, macroalgae is also becoming big time. People are doing seaweed farming to make a lot of money through agar harvesting and carrageenan production. There are specific carrageenans called kappa carrageenan, beta carrageenan, iota carrageenan. Depending on the sulfation pattern, you need in tons of quantities for industrial applications as well. And another thing that's my favorite is antifreeze glycoprotein, right? Poor fish, as we know, they are cold-blooded, right? If you're swimming at -30 in Arctic, Antarctica, your body temperature is the same. Have you seen that supercritical fluid or a metastable water bottle, right? You keep it in the freezer, it remains a liquid. And then you tap it, suddenly you find ice all over the bottle. Have you ever felt that experiment? It's a metastable state, right? But imagine if a fish that's swimming at -30 can freeze, then how can it survive? So they actually have something called antifreeze glycoproteins, which catches the first ice crystal and prevents it from crystallizing any further. Same reason why we add glycerol to E. coli stocks. Same reason why we add DMSO and other things to cell culture and slowly cool them at 1 degree per minute rate, so that those icicle swords do not penetrate the cells. So if we can figure out antifreeze glycoproteins, maybe I could be frozen for a long time and I can come back like Captain America after 70 years, right? Okay, so this is futuristic. And so there is so much to learn from the existing thing. I don't have to engineer freeze glycoproteins. I just have to learn and understand what are these antifreeze glycoproteins happening in fishes and the whales and the sharks that are actually swimming at below 0 degrees Celsius. And then antibiotics from marine sources, enzymes that function at high salt concentration. It's not just carry-mollis all the time. Thermostable enzymes and Taq polymerases, but we can also find high salt concentration enzymes too. And then here is an example, the article that I found in Nature Communication. This is a superb article. This was amazingly simpler language, It is much easier to read than a newspaper. I highly recommend to all the undergrads to read this, where they talk about how to make space exploration, but also sustainable. Sustainability is the key. Okay, when people go on hiking, not so long ago, Everest was full of plastic. I went Everest on Alps. They said, take your apple waste with me, okay? Because those birds are not used to your apples. So we need to have that kind of awareness and conscience to maintain the sustainability. And here it says, while a person can be roaming around in the main cabin, you need life support system and environment control. And the CO2 that they exhale in the space station is actually converted to methane and water and through a hydrogen reaction called Sabatier reaction. And the methane is allowed to be burnt in the space. So they're thinking how to catch that methane also. Okay. So these are the things, each module has to be sustained. And so this is Rakesh Sharma. Some of you may not remember, but he was the first one to go in the Salute one with the Russian collaboration. And now currently Sunita Williams is in the space station. So this is an amazing article that I learned a lot about how to do sustainable. How do they get water from? Are you not supposed to feel thirsty in a day in space station? You need enough water to go there. And the atmosphere doesn't have, it's not like you can catch. It's low temperature, but it's not, there is no water molecule there. So the atmosphere matters. That's the reason why Moon may not have rains or Mars may not have rains, because there is no atmosphere. Earth is blessed with an atmosphere. Okay. And then they also talk about technology readiness levels, where we keep saying we have this technology, that technology, but the moment you ask TRL, we are always stuck at the level of 2 or 3. So very few technologies, even by NASA, are at the level of cellular fungal food production is very good. But if you go to water harvesting and other things, how to recycle waste product in a space station, the technology is still not up to the mark. So we have a lot of catching up to do. And that means there's a whole lot of work for the current generation and the future generations. Okay. And so this is the just a taste of what is the opportunity for bioeconomy and how to understand how to change into a petrochemical economy, into a bioeconomy, at the same time not harming the environment, at the same time being extremely smart enough to take advantage for our own economic benefits. Okay. And all of you believe in Avengers, right? Now, how many of you think they exist? So you believe in Avengers, you believe in dragons. Okay. Then I thought, whether it's mythology or fiction, who cares? At the end of the day, we need imagination, right? What we are without Alvin Toffler's Back to the Future? Or what are we without Isaac Asimov? Or H.G. Wells' Invisible Man, right? So we need imagination on the other hand, which is very important to push the science. And you might as well try what Vishwamitra tried to do with Trishanku, right? Hey, I'll make your own heaven. Be happy there. You not only get water, you don't have to worry about water and plants and Sabatier reaction. You'll get everything there. So maybe in the future, we should think of making a Trishanku Swarga. And even you see this, even the Arabic literature also was fascinated by these stories. And here they show, and maybe we could do one better and say, poor Trishanku is upside down. Maybe we could make him straight as well, right? So it's important, my message is simply knowledge and intelligence combined with intuition and imagination is key to advance human endeavors, be it space or the marine. I will stop it at that, and thank you very much for your attention.
Thank you, Dr. Gopalan. Now I request all our esteemed panelists to please come up on the stage. And we open the floor for questions, comments, thoughts. Please, a show of hands will be very, very useful for us.
Yeah, yeah. Yeah, hello. Yeah. Yeah, I'm a doctor. Yeah, yes. Socrates, from the Sri Venkateshwara College. First of all, I would uh, thank you all for organizing such a good event on the BioE3 policy, and thanks to all of you for introducing the theme of the same. I have a question. Uh, do we have checks and balances introduced with this policy? What to do, what to not do, what what are what are the restrictions? Since it's a vast area, right? There could be more useful means on also, and there could be some negatives also, right? So that's my question. Thank you.
It's absolutely right. Can you hear me? Is this working?
Yes.
So very glad to see Venki here. I'm an alumni from Venki, and it's great to see कि हाँ मेरे और गुरु भाई भी होंगे. So of course there are, with any policy comes the do and don'ts for the BioE3 policy, because of course, does anybody know what are the 3 E's in the policy? Absolutely. So the vastness of the 3 E's, correct? Economy, environment, and what's the third one? Absolutely, employment. You have do's and don'ts for each kind of sub-genre that you heard us speaking about. So within AI, I know that the use of AI on one hand is very helpful, but you have to have checks and balances about what not to. And there is ethics, there's ethical AI as well as ethics for AI. So we in India are part of several international global consortia, such as, for example, the COERA, ERIP, AI for Research Assessment team, in which we are building policies and white papers for the right do's and don'ts for every kind of AI that we use. So that's just one part of it. For the whole policy, I would say you can't have a single sweeping do's and don'ts protocol, but for every subset of what we are doing for the bio-co-foundries, for example, for the bio-AI, for the gene editing, India missed the GM bus. We don't want to be missing the gene editing bus. And what Naveen told you about how the law, the legal system, judiciary in India approved for SDN 1 and 2. So there are do's and don'ts, and we are taking care of them in genetic engineering. Would like to take.
So yeah, so Gitanjali said it right. Especially when I talk about agriculture, in agriculture, we know that genome editing is playing a very important role because it is the most easiest way to manipulate a crop. It's not like ethical clearance, but like human animals, which you need. So crops has very, I'll say, very relaxed guidelines for SDN1 and SDN2 kind of genome editing. So in genome editing, we have categories like SDN1, SDN2, and SDN3. There are 3 major categories. And the RCGM, which is the regulatory body of DBT, Department of Biotechnology, they have laid out very nice SOPs, that is standard operating procedure, what data need to be compiled, how the genome editing crops can be taken to field conditions under your surveillance, under institutional biosafety guidelines. So they're very well worked out. So those are clearly very well worked out for any genome editing crops. Unlike genetic modification, where we keep on even for 20 years, for example, GM mustard, we know that is one of the very good example. DMH11, which for the last 20 years it has been struggling to see the light of the day. And unlike this, genome editing will be, I think, will be a very good option, or it's a very good strategy to take our technology far in a faster manner.
We have a question from IGIB at the back there.
Good evening to all the panel members. So my name is Anvesha and I'm from IGIB. My question to you is that this talks about there are 3 E's like economy, environment, and employment. So the third E, that is employment, when we talk about employment in science and scientific field of research, So how do you think that this policy will be instrumental in changing the employment opportunities in scientific research at all levels, not only just after PhD. It's also about after undergraduation or at the post graduation levels, or after PhD or at the postdoctoral research level, as well as do you think that this policy will be instrumental in preventing the brain drain that is most prevalent in scientific like brain drain in the scientific community?
Audible? Yeah, if I understand correctly, how is BioE3 policy going to help with employment sector, right? Employment sector can be defined in multiple levels, right? Quality of employment also matters, right? Okay, so when you talk about quality of employment, the question to ask is, if you take IT industry as a parallel, right? Every IIT undergraduate, right? BTech students used to be taken up, not IIT or even engineering colleges used to be taken up by all the IT and telecom companies because they needed that much quantity of manpower. Okay. So when it comes to biotech, similar thing can happen, should happen, but it's a chicken egg problem, right? Does the chicken go first or the egg come first? Which comes first? Okay. If the industries flourish, then obviously there'll be need for undergrads to be employed, right? But in order for undergrads to be employed, industries have to foray into this. So compared to the telecom or the IT, which are mass consumer products. Okay. If you are making cell phone, you have to make it for 140 crore people. That's it. Okay. But if I'm making medicine, I need to make only for 10% of those who are sick at a given disease. Right. So we cannot simply take it as an employment. We have to qualify that with quality employment with qualified packages also. So we need to develop skills that can develop. And that is where DBT and the government is also focusing on startups, where you have a fancy idea, but you have that you can actually do with a proper plan using biotechnology. You might want to try it. It may be a failure, right? Silicon Valley had many more failures that we don't hear about. We hear only success stories, right? So the idea is to be able to take risk, and also some of people have to become employment givers rather than employment seekers, right? So if you have a startup on your own, like IT parallel, you will be able to make your own companies. You may be able to employ people. So there are perfect numbers don't come right now. It's not a petrochemical industry. It's not a railway department, right? Where you need manpower to run the show. If you see, for example, banks have completely cut down on the manpower. Where there used to be 20 people, 30 people per branch, run with 2-3 people. Because no one goes, everything is digitized. Okay. So I don't think we can put a quantity number and say this many employment will be generated. It's a chicken egg problem. But quality of employment also matters. And Dr. Mohanty will add to further.
Okay, thanks for asking this question. The minister had a press conference yesterday, last Saturday. This was the question asked by one of the journalists, and minister and secretary of DVT had said that in next 6 months, some roadmap will come. So as he rightly answered, you should not look employment only in terms of a university professor or a scientist. The employment will be there in startup employment. There will be things, some will be high skilled jobs or this one, but policy has just been announced. Within next couple of months, they will come up with some sort of a projection in which areas the employments are likely to come, and some figures would soon come. Okay, thanks for asking this question. Yeah, please.
Thanks, sir. Yeah. So, hope I'm audible, right? Yeah. So, hi, sir. I'm Alok, and currently a research scholar at IIIT Delhi. So, and other than that, I'm also running a startup for the past 2 years. So, I have a question basically that in our country right now, if I have been, if we can see basically that there is maximum unemployment in the field of biotech. We are just keep on increasing the number of MSc biotech graduates, then UG students, but these numbers getting converted into the jobs, the number is very, very and very less. If we look at the some of the companies in Bangalore or somewhere, these students completing their MSc biotech getting the salary of 10,000 per month. getting some internship kind of the things, then few are getting 20,000 per month. So how are you going to justify this? Because every student is not able to afford to go for a PhD. So how are you going to justify that inequality in the education level with the employment thing, and how this policy has the potential to bring that gap or bridge that gap basically? Thank you so much.
So just to add, I will come back to your point about employability. Employability is a larger issue. Okay, that is applicable for every commerce graduate. That's applicable for every literature graduate, every engineering, every medical graduate. Employability is not for granted. Okay, employability by large-scale companies has to come from the training. Okay, if the graduate is not suitable to be employed, why should anybody employ? That's question first. But coming to an earlier point about job creation and becoming startup. So if somebody says, I have a great idea, but I have a thorough plan, and I need to make a startup, and I do not have anybody investing in me, that's where DBT and the government of India is going to say, BioRack, there is an option. There will be mechanisms where we will be able to invest people who want to take risk. But that also will require some. We'll come to that, sir. Okay. So the idea is, I just want to say, if anybody, that's the whole idea of talking to undergrads. So you may have great ideas. You may be the next Steve Jobs. You may be the next Bill Gates, or you may be the next Craig Venter. Okay. So if you have an idea, and then if you're looking, nobody is funding me, but the government is going to say, no, all the capital expenditure we are going to invest in bio hubs, where you will have all the infrastructure. You will have the instrumentation system also. Okay. Like if you need to start an IT company, what do you need? You need a work office, right? You need a good internet system, you need servers, and you need workstations, and then you start your company. But same thing, if you want to do a wet lab, genome editing or genetic engineering lab, you're going to need fermenter or other things in that. So the idea of biohubs are precisely that. Can we provide that kind of facility where anybody with an idea can walk in, spend 3 years, generate a product, and get it bought by big pharma or other people, and then you are sold out, in licensing or out licensing, and then you move on. Okay. But coming back to the employability question, the employability or generation of employment also involves a lot of private sector, right? So the question is, all of us, when we grew up also, we know that this degree will help me in knowledge and ability to troubleshoot things, right? Every BTech guy recruited by Motorola and other IT companies, they got trained by the companies. Okay, they are not ready. Okay, you got a degree, come and start working. From Tata Communications, Infosys, all the IT companies, without having specific names, is that all of them give in-house training. So we need to ask a question, how many biotech companies exist that are both wet lab and dry lab, that have such a demand, that are ready to train for their day-to-day requirement, so that their goals, which is of making profit, can be achieved. Okay, so there is an employability challenge, which would need to be curriculum. It's a chicken egg problem. And it's also true that our biotech industry is still catching up. That is the whole idea of government being the biggest investor in this case. Like highway authorities, right? Highway authorities will put amazing roads, not Ola, Uber, or the taxi guys. It is government investing in infrastructure. I will pass it to Gitanjali.
I just want to say that India has a national education policy, and that policy came in solely to ask the question our young friend asked just now, and that was about how to make people employable. All of us, these numbers of graduates that are being spewed out of all the colleges, how do we make them employable? And employability is therefore, as Gopalan said just now, different from getting a degree and being educated. And so the national education policy decided, it came up, the whole discussion was, the problem is that in the education sector, we are being trained to pass exams or get something, or maybe think of a sarkari naukri, but we're not thinking about what the industry, what the country needs, and what are the national missions. And therefore, there is a need to connect industry and academia. Then since the national education policy, we've been working on connecting industry and academia to overcome the, to answer the question that you asked, my dear, what is it that can make you employable? And now the BioE3 adds to that by saying that through biotech, a skilled workforce in so many different subsectors is going to be generated, adding to the original idea of the national education policy to create people, as for example, through internships, to do things where you're going to become or make yourself more employable. And that is one thing where I believe that the brain drain also can be avoided, because if you're employable, if you've got ideas, if you've got innovations, as we have planned in the BioE3, then there is a futuristic idea that people won't be leaving the country to get, to get that quote unquote feather on my cap, which will make me employable back in India when I return. You would be employable beforehand. So that's the unless.
I just wanted to add up something more. So let's place our GERD, which is called Global Index of Research and Development of our country versus what is there in US or China. So on an average, we talk about investment from industry in China and US especially, it's almost about 70% of their whole R&D is being governed by industry. So they know that, okay, their industry needs this kind of workforce, this kind of investment is needed, and these kind of research mandates are there. So almost up to 70% of their R&D is being supported by industrial grants. Contrary to that, India is right now, we are about 64% is by public investments. So that is, we are mostly from central government or to state governments. Most of our R&D is being funded by that. So the scenario has to now if you have listened about ANRF, that is now we also call this ARF, that is Research Anusandhan Research National Foundation. Research foundation. So it is more oriented towards having more industrial grants. So that is about, so it's about 70% of our funding for R&D should be from industry. So if that could be the scenario, if you can reverse this kind of funding scenario, possibly we are going to create more jobs, more opportunities, more for BioE3 kind of employment could be generated. That is what we are presuming right now.
Yeah. Even in bio-AI hubs, I would imagine, let's say, somebody has an idea. I want to make Taxol in E. coli, right? Yeah, sure. So then you should be able to go to the AI hub and get that data and then do a synthetic biology with all those skills that IT people have. Yes, please.
I have a question. My question is regarding myself, Saksham from Sri Venkateswara College, pursuing life sciences third year. So my question is regarding the biomanufacturing hubs, which are going to be there. So the question is, how will these be hubs like distributed across India? Like, what will be the criteria for selecting the cities, the tier 1, tier 2, tier 3 cities? How will they expand and everything?
Just been announced a week back. As it happens with any policy, the implementation details would come slowly. Okay. So at this stage, even the minister or the secretary will not be there. As the implementation details will come, so these things would come, how it will be distributed. But those of you who are following this area, BIRAC has already called for proposals on biomanufacturing, even before this policy came. So there is that biotech industry as research assistant they give to support industrial research. They have been working on many of the overlapping areas and biofuel and anything. They have been supporting projects, and there are already calls for project proposals and all. And how the manufacturing hubs, where they will be located and how they will be located, this will come slowly. And it will take a little bit, couple of months or something. It will come later. Okay.
Yes. There.
Good afternoon, gentlemen, and the esteemed panel. My name is Sri Babu, and I'm a second-year zoology student of Hansraj College. And listening about the talks on the bio-AI, right? And the probable implications and building like biochips and all. So I wanted to ask, like, what are the potential applications of AI in decoding and stimulating the entire human brain? And how would this advance our understanding of consciousness? So can you please shed some light on this?
हाँ से question पढ़ रहे हो. But the thing is, I'm not a neuroscientist. I do know that the mouse brain is being mapped in a spatial or a spatio-temporal kind of mechanism, where through slices, every bit of the brain, the cells, the neurons, and their interactions are being created to map into a bio-brain kind of scenario. We can think about, in a futuristic way, not an apocalyptic futuristic way, that the human brain may also be mapped in a similar way. And it was somebody's brain that they decided they're going to be this mega, was it Hawking? But it was decided that it would be mapped. But of course, we are very far from mapping. If you could potentially map every aspect, every part of the brain, and know what those neural cells are essentially doing, we have already been able to map it through traditional biological methods, but AI can enhance that by the sheer fact that AI can handle much larger data. Does that answer your question?
I guess, yeah. Thank you very much.
We'll take the question from Dr. Renu Bobade.
Yeah. Yeah, good evening, everyone. Am I audible? Okay. I'm Dr. Renu from Department of Biochemistry, Shivaji College. First of all, I would like to congratulate all the panelists for this wonderful session on the BioE3 policy. Well, my question is to you, Dr. Gitanjali. This is regarding you were talking about some internship program and students were doing internship in your lab. So we have our students from undergraduate and postgraduate. So I just wanted to know what is the procedure and is it like a kind of a training or do they get some stipend also? And for how long they can work in your lab?
Thank you, Dr. Renu. Brilliant question. And I was hoping somebody would ask that. So within Semantic Climate, we are able to get funded by climate-oriented bodies like the UNDP India, like the CoData India, as well as international, as well as several Cambridge and European partners who we have. Semantic is a global team. And so when you do an internship with Semantic, it can be stipended. But in research institutions, not where I belong to, NIPGR, we have been having a tradition of internships for the last, I've been working for 20 years. And I know that for the last 20 years, at least, there is this inter-academies partnership fellowship, internship program called the SRFP, the Summer Research Fellowship Program. It is a paid internship program that is central and national, where undergraduates and postgraduates they choose the labs all over India. So there's a list of all of us are part of that, hundreds of labs. You choose the lab where you want to go, write an SOP, you are selected, you're paid to travel. Otherwise, if it's in your own city, whatever else, and it's a stipended internship. Similarly, individual state science and technology councils also have paid stipended internships. For Semantic Climate, which I run, we have paid stipended internships when our national or international partners are funding it. At the rest of the time, because we are a global initiative of volunteers, we run it all the time constantly. At NIPGR, we run 6-month internships, which are for postgraduate students, but within Semantic Climate, we do it for undergrads who have summer mein chutti and that time alone. More often than not, undergraduates will work with you for 2 months maximum. Yeah. And that is the time that we provide. It's not just when I say me, it's not just when I say we, it's not just me. It is the 14 institutions of the IBRIC, which are in India, which are providing such internships, and different projects under modules are providing internships to people with different levels of stipend. Somebody mentioned कि postgrad करके भी आपको 10,000 का job मिलता है, but the stipends that we give in Semantic are starting from 25K for somebody who's just sometimes not even we've had school kids on this also. Anybody who's interested to help us to do, we have those programs. But maybe, Shams, you have in biomanufacturing a much better scope for internships for students to come in, and maybe at ICGB with the international program, you would know better about other avenues for internship that you might want to tell our students.
Of course, the internship we do very regularly for all the stipend thing is, as already said, it comes from the Indian Academy of Science program, that summer fellowship program. So the students apply through that, they choose the lab, they have a proposal there, and we get the final selected students, they get stipend for 2 months and we work with them. And they get into the biomanufacturing program. Most of our lab in ICBB are dedicated to biomanufacturing. And in fact, I must say that this BioE3 is something that pushes biomanufacturing, right? I think this is the first time at such a large scale, Indian governments are pushing biomanufacturing. Earlier, biotechnologies were there. There was research, R&D push was there. For the first time, so much of biomanufacturing. And that is employment. Questions were coming about employment. I mean, if you have so much of push from government to go beyond the lab and get into the biomanufacturing, you're automatically going to have an employment. So yeah, I mean, the younger generations, I think the new ideas and training, they're all there for training purposes. They're all available. So whenever you get it.
Yeah. Thank you so much.
We have 2 questions here from Zakir Hussain.
To add to that would be the scale. We still don't have the scaling capacity. If you want every one of your student to benefit from such training, then we have limitations as well. So scaling also should be thought of. Hopefully we know better.
Good evening to all. So my question is, there is economy myself Aryan Gautam from Zakir College of Botany Department. And my question related to that word economy is there. There are some areas which are important, but they don't cash in as much as money, such as neglected tropical diseases. And it also affects a very high population, such as 1 billion of population is affected by neglected tropical diseases. But pharma companies don't make specific drugs for it, such as sometimes for the diseases related to protozoans, they use malarial drugs for it. And it affects population, and it also increases fatality towards them. 40% of population gets affected by it. But there isn't any major research, or if there are researches, they are being neglected by pharma companies. So how can these policies help to solve this problem?
Hello. Okay, so to address this, what is being done at the government level is, of course, there can be situations where there are, the neglected disease being one of it. You could also have diseases which are, just showed up on the scene and caused havoc. So on to kind of cover for this, what the government and the DPT has been doing it through various missions is to try and fund some of these areas so that we can take the research and the necessary products. For example, if you were to think about chikungunya and you say, okay, we have, let's try and make a vaccine. It may not have gone through all the trials, but you have taken it far enough that in case you have a very big pandemic, you are very close to actually bringing it up in a manner that can be utilized. So there is recognition for this kind of, areas, and government is supporting these efforts. And again, they may not be, much scale, so to speak, and the number of people who may be suffering with some of these kind of diseases may not be huge, not all put together, but in numbers. But nevertheless, it's being recognized, and efforts are being made so that you are well poised to take it up in case a huge emergency comes up.
So, good evening, all the panel speakers. My name is Harshita Tripathi, and my question is that how can we, as a college student, contribute to the BioE3 policy?
Is this
Nice question, actually. So the way you can contribute is start thinking. So there's the ideathon. I think Department of Biotechnology has already put up on its website what is called ideathon. Can you give new ideas? You will become automatically part of this policy if you bring out new ideas how to make this policy successful. See, somebody asked about do's and don'ts. I think what is important is doers. Do we have doers? I think if this policy will enable people to become doers, that would be the success of it. I mean, don't really come later on. First, let us do it. Policies are there everywhere. We keep on making policies, but do they really work? We only make it work. So you are the younger generation. You are the people who will make this to work. So think about ideas, how to make it enable.
I want to say two things, what you can do. One is remain aware. Ignorance is not bliss. Whoever might tell you it is not. So be aware of what are the opportunities. For example, one of your teachers asked me, internships kaise karte hain? None of you thought about asking. Online internships hote hain, offline internships hote hain, BioE3 ke specific area sectors mein hote hain. You guys should be aware kahan ho raha hai, kab ho raha hai. Awareness is one thing. The second thing is hold on to the passion that you have. I see youngsters have so much passion about and interest in an area, but as you do your senior studies and get into a job and then you tend to lose, and somehow that passion somehow gets lost because you get into the stumbling day-to-day routines. Try and hold on to the thing that you like very much. Rather than if I tell you ki aaj ka best, you want to do AI, I want to make you do AI. It's great, I believe that because I have a passion for it. If you don't believe in that, don't do it. I'll give an example. A student of mine came to me कि मैं botany करके आया और मुझे पोलन को देखने का बहुत शौक है, तो मैं पोलन को ही देखूंगा यहाँ पे. And I said कि नहीं, इसमें AI कर लो, bioinformatics कर लो, कुछ कर लो, उसको पोलन को देखो. And then it turns out that at IIT Delhi, there's this guy who has invented a different kind of microscope, a digital holographic microscope. So I said, चल देखो पोलन देखना है, जाके IIT में देख के आ उनके microscope के through. And this boy went and looked at it because he knows how to handle pollen because he loved it during his college. The botany teachers made him love it. And so he looked at pollen under the DHM and he was able to see, and we discovered to our shock, the viability, जो कि हम हमेशा से staying 70 साल से हम लोग viability कैसे access करते हैं pollen का? Through staining, right? And this guy found that the DHM allows you to see and identify viable versus non-viable pollen straight away just by looking through the microscope. We published it in quantitative plant biology. We were interviewed about this, ceiling crashing idea of looking at pollen and being able to identify. And I said it was not me. It was the student who had the passion about wanting to do what he wanted to do. And that's something that you guys only can do, right? I hope that makes sense.
Yeah, I would like to add 2 points quickly to the earlier neglected tropical disease, right? One of the things that we don't have to wait someone else to do research on our diseases. And also, I would like the patient activism to be there. Okay, there may be 1 in a billion that's affected with a certain disease, but the patient and the people should also push for it, right? And of course, somebody will be doing a little bit of research publishing here and there, but you want to cure the disease, you want symptom management, things like that, right? So we need to create awareness to the lay public as well, that this is a disease happening in this particular geographical zone, and we can do many things using the state-of-the-art biochemistry, biology, that drug development that we know. It's simply a matter of getting it done. So we need local activism also. I know people like who would make a foundation for, vitiligo or autism disease simply because they are feeling it on a daily basis. They have someone in the family. So things like that have to happen. So there has to be larger effort from the public who is suffering to say that I'm not a scientist, but I want this solved, right? So the patient advocacy group should also actually evolve. And in terms of what undergrads can do, apart from what Shams and Gitanjali said, you can become an ambassador, right? If you go to a marriage party or anything, or on your WhatsApp group, if you see some of your little baby nephew or niece using Augmentin, find out, tell them what it is. Tell them how is it killing bacteria. Tell them what penicillin is about. Tell them what CRISPR-Cas9 is about, okay? And any medicine that you take, learn about it and spread. It's not like everybody in the country should know science, but if you convey to them in a layman language, which I try to do myself, would be very helpful. Okay, tomorrow they may decide our funding, so it's better to pass on this science. Like when we lined up for oxygen for COVID, we have to understand that O2 is the one that saves, right? Not N2, right? So you have to be an ambassador as well, and you can help a lot in terms of general scientific awareness. And BioE3, of course, you have to be the enabler. You have to be the initiative. All that BioE3 says is that you have an idea, you want to execute, you are looking for infrastructure, here is the infrastructure. And that would be cool if you can rent for a year or two and then make a great product, right? Thank you.
Hello, ma'am. Hello, sir. My name is Nishtha Bhatnagar. I'm from Maitri College, BSc Life Science, third year. Yeah, so my question is regarding BioAI. So coming from a BSc biology background, we don't know much about technology and technical languages. So where do we start? How can we start now to get into that field tomorrow? Where to begin and from what?
To understand BioAI or to understand AI first?
To understand BioAI.
Or to start working on it because you feel you want to be.
Yeah.
So to be able to get into something that you want to learn, what I have found in my life is I learn best by teaching. When I don't know anything, I start to teach it to youngsters. And like Gopal was saying, even if it begins with your own family, start teaching something. But of course, as of today, professionally, if you want to pick up a new skill, let's say you want to understand what is artificial intelligence, how is it different, for example, from machine learning that we were doing earlier, or hidden Markov models that we were doing earlier, you need to understand those models first of all. It begins with data, and the data, what is data? It is data science. So is your data, think about any data that you have in mind, all the people in this room. What is the data we know about the people in this room? Are we thinking, do we have a programming logic? So yes, these are people, individuals. Can we categorize them? Colleges, yes. So is this continuous data or is this categorical data? If it is categorical data, what can you understand? How can you model it? If it is continuous, how else? So you have to learn something about data science first. When you go on from data science, you can bring on different kinds of transformer models. How are transformer models doing better than the machine learning models that we had in the past? And so therefore, supposing आज की date पे you yourself decide कि मैं घर जाके अब कुछ AI पे start करना चाहती हूँ, I would say find some basic tutorials. Never a 6 महीने वाला, 3 महीने वाला. Think about lectures that sometimes people give, short lectures. Many of us have done it on YouTube. And YouTube is the ultimate free place to get everything under the sun. Listen to a couple of lectures, just listen without wanting to learn or make notes. Listen to something, you will get a hang of your head, you will wrap around the basic terminology. Then go for a half-day course or a one-day course, which you could find freely on Coursera.
At least for AI, there's the University of Cambridge, the University of Oxford, MIT are creating these courses. Amazon Web Service has made so much free. वो चाहते हैं हम उनसे खरीद लें, of course, surveillance capital. But Amazon AWS has made so many courses of basic data science and AI freely available to anybody who is interested. So that is another thing. So I'm assuming without paying for these things, I mean, you should be able to learn something as a student for free. So there are so many courses available out there. YouTube pe bhi bahut sare. But then there is again this information overload. You get 10 zillion courses on one topic and you don't know where to begin. Begin with single lectures. Try and attend more events like this. Thank you for coming today. I think it's very important for us also to get to know you, the kind of questions you're asking, so that next time we prepare our talks from, build them from that point of view. This is what students want to understand. This is where we can go. So begin with short and then scale it up to whatever you understand. Read, and read some more.
Ma'am, I did learn database management system, DBMS, but I don't know how to implement it in biology.
Oh, right. It's very simple. So DBMS, for example, we are building phytochemistry databases. So you want to know contextual, there's the Earth Metabolome Initiative. In the Earth Metabolome Initiative, the idea is entire Earth, whatever living organisms are there, we are going to profile their chemistry, right? All of life is complex chemistry in a semi-permeable bag. Jo khushbu hame aa rahi hai, basically it's coming from wherever we are. Himalayas mein jao, immediately bluish tinge ho jata hai. That's because of the monoterpenes of the pine trees in the Himalayan foothills. Chalte hi pata chal jata hai, ab hum plains se nikal gaye, Himalayas mein aate hain. So the whole idea is when you have these data sets, you put them in a fair format, findable, accessible, interoperable, reusable. You cannot put them in a fair format in a PDF file or in a table on an Excel. You have to put it into a DBMS. And that is where the DBMS is what allows you to do browsing, searching, asking queries, making combinations, doing stats, doing data science becomes possible when your data is saved in a DBMS. So I'm glad you picked up a DBMS. From the DBMS, there's so much to do. I'll talk to you later about this. There's so much you can do.
The
Thank you so much,
best thing is you apply for summer internship in Netherlands now.
A question.
Good afternoon, everybody. My name is Vivek Bhardwaj, and I'm pursuing biochemistry honors from Sri Venkateswara College. So my question is from Sir's part. Like Sir said, plants only optimize 20 to 30% what we provide them. So what is the root cause of that? Like plants only take up 20 to 30%. Why not more? And how are we going to tackle that? And if we generate some kind of plant which can gain more of the percentage what we provide, then how the government or the institute responsible for generating is to reach the farmers, because in India, accepting a new thing is very tough, and especially in the rural area. So what are the plans for that?
You see, if you go through NIPGR website, and I'm sure you are going to get a lot of information about this. Broadly, if I talk about it, because I don't work specifically on those nutrient use efficiency stuff, there are many good labs in India as well as in NIPGR also. So there are three major micronutrients, if you remember. There are nitrogen, phosphorus, and potassium, and PK, which we call. So plant do take them, but mostly as a inorganic or organic, that is something that has always been known. But what we know that the inorganic form is not so readily accepted, and then the plant is unable to take it very efficiently because the plant has some limitation in terms of its anatomy as well as its uptake in terms of the enzymatic reactions which is there in the plant. So there are major areas, challenges to kind of manipulate these pathways which can have both uptake from the roots as well as also to assimilate these for their further conversion, because it's not that once uptake is taken then it has to be efficiently kind of taken up. After taking it has to be efficiently taken for some of the pathways, some of the secondary metabolism, primary metabolism. So all these things has to go in sync. So how, for example, if a phosphate is taken, whether it has to be mobilized through xylem or phloem, and how it can go further, that is being done. And I'm sure if you go through NIPGR side, and there are many labs internationally also who are working on these areas, because this is a very important area. If you look about the subsidiary on these agrochemicals is huge. We are importing and kind of putting a lot of economic budget in our for these fertilizers as far as NPK are concerned. So please go through it. I'm sure you will get a lot of information about it.
We can take one last question there.
Good evening, ma'am. My name is Shreya. I'm a third-year biochemistry student from Sri Venkateshwara College. So one of the focuses that I guess was mentioned in BioE3 policy was precision biotherapeutics, I believe. And as ma'am said, that in today's modern world, every day there's something new that's coming up. Even in storage from like floppy CDs were in our childhood. It's so strange to think that it's like not usable anymore. But like a lot of the newer generation, they don't even know about it. But yes, we have cloud computing as the new solution to this large data problem. Not really a problem, but the yes. So I was wondering how the BioE3 policy aims to actually integrate this large flux of healthcare data that we are getting in India, along with this cloud computing, if they have a plan for that, because I believe that it would be key.
Okay, so see the data cloud still stores the data somewhere. Okay, so earlier you stored the computer on your computer's hard disk or on a large storage server on your institute. But what has happened is cloud computing has taken this server elsewhere. So they put the big data center and all this, they store it there. So right now what is happening is there are some people you can either buy cloud computing services, but there are, when it is health data, there are some restrictions because there are some personal information and all that. So government has some rules that you cannot host this data outside the country. There are this. So there are cloud provider, they are certified by the government, like even AWS, Azure, Google. So they can do that. But in addition to that, what government is doing is we are building our own data centers. Like if you studied some, I'm sure bioinformatics, all of you study, like you know about NCBI. So that is a biological data center located in USA. EBI is the European, this one. Same way, the DNA data bank of Japan is there in Japan. So in India, earlier we did not have any primary data center. Okay, there was data, they were scattered in NIPGR will keep their own data, NII will keep the data, other big universities will keep the data. But now what government is building is Indian Biological Data Center. This is has come up in Faridabad National NCR Bio Cluster. So this will be something, a resource similar to NCBI or EBI and all these things. And those things in future would be available to people like a cloud. Like you can access that data from your college or from the research center or something. So this will be a something like a hybrid cloud. You can say that certain type of thing you can also merge with it, like Amazon cloud or that type of something which is less sensitive. You don't care where it is kept, but there are certain things which are more sensitive data. You still you keep it on a restricted place, and you have more restriction who will access it, how will access it. Even in NCBI, there are many data which is relating to patient information or something. You don't get it like free. You sign some agreement. It is not only you, your head of your institute has to certify that it will be used in an ethical way. Only against that license you can access it for this one. So same way, this sort of like data centers, which will of course expand in scope, that will take care of all these requirements. But certain data, which is probably not, that can be put in open access, probably could be put in cloud services, which any institute, research institute will subscribe or something. Okay, does that answer your question?
Yes, sir. Thank you.
Just wanted to add a little bit for a lot of information regarding the hospitalization, how the data is taken, what kind of information is gathered. They are being tabulated. It's just started in some hospitals. It's picked up in a certain fashion, which then is put in databases that can then be queried. Okay, and one could think about what kind of symptoms were there, how long did it take to cure, what medicines were given, what was the age of the person, which part of the country is this disease prevalent. So these kind of information can then be put together in a manner which is kind of anonymous in a sense, because privacy issues are involved, and then they are being one may be able to come up with situations that can, you can come to the diagnosis much faster, and also places where you don't have access to hospital facilities. Some of these kind of information can be useful in, at least providing some help to people where hospitals are not there. So it's on the cards, it's happening in some places, and in the years to come, it's going to be more prevalent.
Thank you. I think that was the last question of the day.
And one last question, ma'am.
One last question? Okay.
Hello, esteemed members of the panel. My name is Rajat, and I am currently a second year BSc Life Science student from Hansraj College, University of Delhi. Ma'am, I was very interested by listening to a speech on the AI. Ma'am, I am very interested in and passionate about cancer bioinformatics. So I have one question, that how can we implement AI in the field of cancer bioinformatics? And that is the application of AI on NGS or drug repurposing or drug design. What is the progress? What is the procedure of applying the AI on that field? And what is the further step at how can we commercialize this idea?
Thank you very much for asking that question. So I had mentioned 2 or 3 different ways in which AI can be used: AI over text, AI over vision. I did not mention AI over genomes, and that is a very, very, very important feature. And expanding more and more. So if you all of you have used AI over text, because I'm sure each one of you has used the ChatGPT. So these are GPT models that have been trained on textual information, not really historical books so much as pichle 10 saal ka available public internet ka data. In a similar way, you can train GPT models on DNA. You can train GPT models on protein. But when you do GPT model training on proteins, it is not ChatGPT, it is ProtGPT. When you train GPT models, you have DNA GPT, which has just been released last year. In addition, there are BERT models. If you want to use a BERT model for DNA, there's also codon BERT. We in the lab are using codon BERT because I work at the National Institute of Plant Genome Research, and I told you we are using it for food security. We are looking at large genomes of plants. And plants for bioprospecting, particularly, we are very keen on identifying these biosynthetic gene clusters. Now, biosynthetic gene clusters traditionally was found, these are the antibiotic making clusters in microbial genomes, right? And there's a huge amount of work that has been done, more so at NII itself, in mining the genomes for these clusters, which would make new kinds of drugs. And then, but that was before AI came in. Now, using codon BERT, using DNA GPT, using ProtGPT, we are in a position to mine genomes at far more accurate scales and far more scalable ways to be able to get. Now, you are saying extrapolate it to cancer. Yes, absolutely. What is it that makes a certain cancer? It's not a one gene, one protein theory anymore. We have the omnigenic model today in biology. And so therefore, it's only AI. No amount of mapping, matching, blasting, clustering, or the traditional bioinformatics mechanisms are going to work with the kind of data we are, the data onslaught that we have seen today. And therefore, it's going to be AI which can address this question. We have, here at the lucky thing about us is if we are not doing the research ourselves, we are in the DC committee of another student with another guy so Naveen is in the DC committee of many of my students. I'm in the DC committee of Devashish's students. One of his students actually is looking at genomic signatures for antibiotic resistance. Now, how would you look at AMR for Mycobacterium tuberculosis, for example? Then we have students in JNU, where again, I'm on the DC committee, and I find that the students are there looking at signatures for resistance or to staph. And these are genomes of variants of so many different strains of the microbe. And you can use AI to be able to find an explainable AI, not the regular. You can use explainable AI to explain or find out what it is in that genome which makes it resistant to a certain antibiotic. In the same way for cancer, depending on which cancer you're looking at, I still have and it's a psychological issue of not being a cancer biologist myself, but I feel there's a lot of lifestyle, the situations associated with cancer. And so therefore mapping cancer would also be something that features of the individual precision medicine, as well as, your gut microbiome. To what extent is it making you a little bit more prone to certain diseases, such as certain cancers? Maybe in the long run, futuristically, again, the BioE3 allows us to close our brains and think about what is the future. And I think there, I hope it answers your question. AI has the scope to be looking at all of these features and to be able to map them, just like in the human brain. So these Ramjas kids are very smart. Okay.
Can I ask a question?
I think what we can do is when we go out for tea, you can interact with the panelists and the experts if they're available for the questions.
Okay, that would do.
Yeah, and with that, we come to the end of this session. Please, a big round of applause for the panelists and experts who have joined us today, taking time out from their schedule to interact with the students. And also a big round of applause for all the Science Setu colleges who have joined us. I just called them up that we are trying to do this, and they said, yes, we are more than enthusiastic to participate in such a discussion. So another round of applause for the Science Setu colleges joining us today. And lastly, a big round of applause for all the students that have joined us today and have interacted with us, because the very fact that you asked a question, how can we collaborate, or what role can we play in the BioE3 policy, is just more than encouraging for us, because the whole idea of putting this together was we reaching out to you because you will be playing a very critical role in the implementation of such policies of India. With that, we come to an end of this session. We'll be open to your questions. You can write to us. You have our email IDs, and we will keep on the interaction and the engagement going. And let's all head ahead outside for a group photograph and a cup of tea. Thank you.
Okay.